on va passer tout de suite à notre deuxième conférencière. Gardez vos questions pour la fin. Alors, notre deuxième conférencière, Juanita Carolina Rodríguez, qui est étudiante au doctorat à l'UQAT sous la direction de moi-même, Yves Bergeron et Stephen Campbell de l'UQAM. Sa conférence est intitulée Forest Type versus Host Identity as Drivers of Moss Phyllosphere. Alors, Juanita, je te laisse la parole.
Merci beaucoup, Nicole. Je partage ça. Mon présentation. OK, vous voyez bien la présentation?
Oui, tout à fait.
Okay, c'est comme, je vais mettre les pointeurs laser. Voilà. Oops. Okay, so my presentation will be in English. So that question are welcome as well in French. So welcome. My presentation is entitled Forest Type versus Host Identity as Drivers of Moss Phyllosphere. Changes in tree canopy composition of boreal forests are mainly due to wildfires, but also related with anthropogenic causes such as land colonization, mining, and forestry. These changes have produced an increase in broadleaf forests, such as trembling aspen, in areas that were normally dominated by coniferous forests, such as black spruce, producing important effects on understory vegetation. Sorry. So one of the groups that could be particularly affected in the understory by changes in tree canopy compositions are the Bryophytes and their associated microorganisms, considering the significant differences of mosses between both forest types and their important ecological functions in ecosystem succession and resilience, as well as in nutrient cycling. So mosses have several bacterial groups in their phyllosphere, but particularly cyanobacteria has been further studied because of its important role in nitrogen fixation, significantly contributed with nitrogen inputs in boreal forests. So as an example, I show here the cyanobacteria nostoc in the phyllosphere, so in the leaf surfaces of the feather moss Pleurotium strawberry. Now, despite all these important ecological functions of mosses and associated bacteria, it is not clear which drivers structure microbial communities in feather moss phyllosphere, because few studies have explored the complete bacterial diversity associated with bryophytes and the factors driving these associations. In previous studies, it has been shown that the phyllosphere bacterial community is mainly structured by moss host identity, which is possibly due to the physicochemistry of leaves or other moss leaf traits that shape microbial communities. However, it has been also suggested that certain environmental conditions could influence moss-associated bacterial communities. So, for example, nitrogen seems to play an important role in defining cyanobacteria associated with feather mosses, because when nitrogen is limited, feather moss can actually secrete chemicals to induce the association with cyanobacteria in order to fulfill their nitrogen requirements. So, considering that black spruce stands are frequently nitrogen limited, the moss cyanobacteria association would be promoted in these stands, contrary to the nutrient-rich deciduous forest. In our project, we quantify the changes in microbial communities as a function of moss species identity with the two feather mosses, Psyllium crista castrensis and Pleurotium strawberry, comparing stands dominated by either black spruce and trembling aspen. These two feather mosses are actually the most abundant mosses in the boreal forest and dominate the understory of black spruce forest, as you can see here in the picture. As a hypothesis, we first expect that moss host identity is the main driver of diversity and abundance of moss-associated bacterial communities compared to forest type. Then, as a second hypothesis, we expected that if forest type as a secondary driver of moss-associated bacterial communities, the overall bacterial beta diversity will be higher in trembling aspen than in black spruce stems. And the abundance of moss-associated cyanobacteria will be higher in the low nitrogen black spruce forest than in the nutrient-rich trembling aspen forest. So this is the study site that is located in the northwestern Quebec, in the Abitibi region, and the north of Rouyn-Noranda. The experimental design consists in 3 different sites comparable in abiotic conditions. Each site has a dominance of either black spruce or trembling aspen, and 2 blocks were placed in each stand. And in each block, 6 samples were collected per moss species for a total of 36 replicates. Then the microbial DNA was extracted. We sequenced the bacterial 16S rRNA gene with the universal primers by Illumina sequencing. And finally, we obtained these sequences that were processed by Data2R package to obtain amplicon sequence variants called ASVs that are actually the equivalent to a species, but for sequencing data. And these ASVs were taxonomically assigned to different bacteria based on database. As results, this NMDS ordination show is based on the bacterial relative abundance of the ASVs, of the equivalent of species, and then points in circles correspond to data from trembling aspen forest. And the triangles in here correspond to black spruce forest with their respective ellipses based on the standard deviation. Now we have moss host identity in different colors, so Pleurotium strawberry in orange and Thylium cristacastrensis in green. So according to the Permanova test, moss host identity, forest type, and the interaction were all significant in defining bacterial community composition. We can also notice in the table that the effect of forest type was twice as the effect of host identity. Therefore, the initial first hypothesis was rejected because forest type was actually the main driver of most associated bacterial communities composition compared and not most host identity as expected. Then, by adding the phyla a posteriori of the bacteria, we can observe which bacteria are related with each stand and each moss, the community composition, and show that there is a higher beta diversity in trembling aspen stands for both mosses compared to the black spruce stands. So the second hypothesis is accepted. Then we wonder how 3 canopy dominants have an influence of moss phyllosphere despite host species identity. So comparing both forest types in the homogeneous black spruce forest, we have inputs of the nutrient-poor needle leaf litter, difficult to decompose and acidifying soils. Then the understory community structure is composed by some ericaceous plants, but is mainly dominated by the 2 evaluated feather mosses that actually are forming relatively poor colonies in the understory. So in contrast, the heterogeneous stands dominated by trembling aspen trees have inputs with nutrient-rich broadleaf litter, promoting nutrient cycling and influencing a high diversity community structure of plant and microorganisms in the understory. In this sense, most colonies are more exposed to microorganisms from surrounding environment in soil and in litter. And possibly there is a sharing of bacterial communities from needle leaves or broadleaves, litter, or even from soil that could influence this moss phyllosphere in each forest type. So as another part of my project, I actually evaluate if there is a sharing of microbial communities from plants or from the phyllosphere of leaves for each forest. And we can also observe that the proteobacteria and acinetobacteria were more abundant in black spruce forest, whereas the Bacteroidetes and cyanobacteria were more abundant in black spruce forests. So if we look at in detail the cyanobacteria group, so if you remember the third initial hypothesis that was based on the assumption of that several articles state that that nitrogen poor coniferous forest would have a higher abundance of moss associated cyanobacteria that's fixing nitrogen compared to deciduous forests. So Okay, so it's important to mention that in previous studies, it has been observed that it's a negative effect of broadleaf litter on feather mosses and cyanobacteria abundance, as well as the related nitrogen fixation rates, which are due to either a physical effect by shading or crushing the moss, or by a chemical effect related with the allelopathic compounds such as phenols. However, as you can see in this figure, the cyanobacteria relative abundance of bacteria associated with both feather mosses in our study was actually higher in trembling aspen stands compared to black spruce, which was contrary to the initial hypothesis. Furthermore, nostoc represented 96% of relative abundance of cyanobacteria and trembling aspen stands. It was mainly nostoc that was present. One important thing to consider is that most of the studies are made of coniferous forests. Just these two previous studies evaluated moss phyllosphere across different forest types, including just coniferous forests dominated by spruce species, open tundra ecosystem with no canopy, and deciduous forest dominated by birch species, including one small tree, betulanana. So the conditions are quite different, and these articles still state that host identity was the most important driver. Therefore, the less studied boreal deciduous forest is proven in our study to be an important habitat for cyanobacteria associated with both feather mosses, and required further analysis of the factors related with tree canopy composition driving changes in moss phyllosphere. So as a conclusion, the effect of forest type as driver of moss-associated bacteria and the significant abundance of mosses in the understory of boreal forests highlight the importance of changes in canopy compositions in the boreal forest. That also then changes in canopy composition would be particularly influenced cyanobacterial moss association that are actually important for nitrogen-limited boreal ecosystems. Now, the reasons as future prospect of how different conditions related with tree canopy dominance shape bacterial association with mosses and the relative performance for the ecological functions such as nitrogen fixation remain to be elucidated. Thank you for your attention. Je tiens à remercier aussi les partenaires RAIAM, Gestion forestière, Norbord, et le financement par MITAX et le feront du Québec. Merci beaucoup.
Merci beaucoup, Juanita. C'était une très bonne présentation, puis un excellent respect du temps alloué.